All right, let's jump right in. We've been reading our genetic code for decades now, right? But what if I told you we've been missing a huge part of the story? Today, we're going to look at a new tool that's giving us a much sharper picture of how our genes actually get their work done.
It all really starts with this one big question in biology. Think about it. A neuron in your brain and a cell in your skin have the exact same DNA, the same set of genes. So, why on earth are they so different? One's firing electrical signals and the other is making your skin. The answer to that puzzle lies in a layer of complexity we're only now, finally, starting to really see clearly.
And the key to unlocking this is a concept called a gene isoform. The best way to think about it is with a recipe. Let's say a gene is the master recipe for making a protein, like a recipe for chili. Well, isoforms are all the little variations on that recipe. You might have a spicy version, a low-fat version, maybe a vegetarian version. It's still chili, but those small tweaks can lead to totally different results in the cell. So if these little recipe variations are so important, you're probably asking, why haven't we been talking about them all along? Well, for a long time, our view has been pretty incomplete, and it all boils down to the technology we were using. For years, the main way we read genes, short-read sequencing, was kind of like trying to read a book that's been put through a paper shredder. You get all the words, sure, but you have no idea how the sentences, the full recipes, were actually put together. Now, long-read sequencing came along with a promise to fix this. It could read the whole sentence, from start to finish, but there was a pretty big catch. The early versions of this long-read technology, they had a really high error rate. So yeah, you could read the whole sentence, but it was like the ink was all smudged and blurry. This made it super hard to be confident what you were seeing, especially when you're trying to read the tiny little barcodes that tell you which cell a specific recipe came from. And then, just a few years ago, a technological leap happened that completely changed the game. Suddenly, the lens we use to look at our genes became crystal clear.
This new generation of long-read sequencers, specifically the Oxford Nanopore R10s, dropped the error rate to just around 1%. 1%. The ink wasn't smudged anymore. The text was sharp. This was an absolutely massive leap forward.
So this amazing new hardware created a fantastic new opportunity, but also a new problem. We now had this beautiful, high-quality data, but all the old software tools, they were built to work with the old, messy, error-filled data. They weren't right for the job anymore. We needed new software, something designed to interpret this incredibly clear new picture. And that is where our hero of the story comes in. A brand new computational pipeline called SCOTCH, and it was built from the ground up specifically for this new era of super clear, high-fidelity data. Now, SCOTCH stands for single-cell omics for transcriptome characterization, and its whole purpose, its mission, is to stop worrying about fixing errors and instead focus on harnessing the incredible power of these clear, long reads to explore all the beautiful complexity of our genes.
So how does it do it? Well, first, since the data is so clean, it can just read the cell barcodes right off the long reads themselves. No other guides needed. Then it maps those reads to all the known recipe variations, or isoforms. But here's the really clever part. It takes all the leftovers, all the reads that don't match anything known, and it clusters them together to discover completely new, never-before-seen isoforms. And finally, it has a really smart way to handle reads that could potentially match more than one recipe, making the right call to avoid confusion.
Okay, that all sounds great on paper, but the researchers had to prove it, right? How do we actually know that SCOTCH is any better than the tools that came before?
So they set up this massive simulation. We're talking over 15 million reads where they knew the exact answer ahead of time. They knew which isoforms were there, how many there were, which ones were new, all of it. Then they basically let SCOTCH and the other tools loose on the data to see which one could get closest to the real truth.
First up, read mapping accuracy. This is basically how good are you at matching a read to the correct recipe. And SCOTCH did great. It scored about 0.88, putting it right up there with the best of them and way ahead of some other common tools. A really solid start. But this next test, this is the real showstopper. It's called gene count accuracy, and it measures how well the tool actually counts the amount of each gene. And look at this. Scotch comes in with an accuracy of 0.77, while the competition is way down here around 0.2. It's not even close. And that huge difference comes from how smartly it handles those tricky, ambiguous reads. It just gets the numbers right.
Okay, so it aces the simulation, but that's a controlled environment. Can it actually find something new, something real in an actual biological sample? Can it make a discovery that matters? The answer is a huge, resounding yes. When they pointed SCOTCH at human blood cells, it found a completely new version of a gene called EIF6. And this wasn't just some tiny tweak. It had a splicing junction, a piece of its recipe that had never been documented before. And the best part? They went into the wet lab and used a standard technique called PCR to prove it was physically there. This was a real, verifiable, brand new discovery.
So what does all this mean for science? What kind of biological secrets can we finally start to uncover now that we have this incredible new level of detail?
Well, let's take a look at a gene called TSC22D3. It's a really important gene for regulating inflammation. Now, using the old methods, if you just looked at the overall amount of this gene in different immune cells, it looked, well, boring. The level was flat. It seemed like nothing interesting was going on at all.
But SCOTCH revealed something fascinating was happening right under the surface. It found an isoform switch. In one type of immune cell, monocytes, this one version, we'll call it isoform A, was totally dominant, making up about 65% of the total. But in other immune cells, the script completely flipped, and a different version, isoform B, took over. The total amount of the gene never changed, just the version being used. A critical secret that was hiding in plain sight.
And the impact of this is just massive, especially for something as complex as the brain. When they applied SCOTCH to lab-grown models of the human brain, it found 15 times more significant gene changes than the other tools. 15! It uncovered these crucial isoform switches in genes that we know are critical for how neurons grow and connect with each other. And maybe most importantly, it connected these discoveries to proteins that are already associated with autism spectrum disorder. This just blows the doors wide open for new research.
So with tools like SCOTCH, we're not just reading the genome anymore. We are reading it in glorious high definition. We've effectively unlocked an entirely new layer of our own genetic code. It's revealed this hidden, dynamic world of competing recipes and isoform switches. And that really leaves us with one big, exciting question. What other profound biological secrets have been right there all along, just waiting for us to finally have a clear enough lens to read them?